To go?
Sounds good.
Okay. Well, good evening, everyone, and thank you all for joining us for Dr. Pfao's presentation. This is Dr. Russell Pfao. He's a PhD in biology at Tarleton State University and is really a genetic studier. That's his primary background. But this evening, he's going to speak to us a bit about how iNat is used by scientists and by science. And since we have this City Nature Challenge coming up soon, that's kind of been the emphasis on what we've been trying to study and learn more about. And um, we thank you, Dr. Pfao, for joining us this evening and giving us your time to work with us and help us understand things. And we thank you so much. And we're very excited about your presentation. I met you a year ago and it's almost to the day. So I look forward to seeing how much smarter you are in a year. And let us know what you have going on. But thank you so much. And without further ado, I'd like to introduce Dr. Russell Pfao. Please, sir, the podium is yours.
Okay, thank you. You asked me to add a little more information about myself, so I'll just start there. Unlike a lot of scientists, I have a really broad background, and my scientific career probably began in my single-digit years. Right after I got out of diapers into regular pants, I became a scientist and have been interested in plants and insects ever since. So I decided to go and get a biology degree, formalize that interest. And I thought that I wanted to be a wildlife biologist working with Texas Parks and Wildlife. And once I got into college, I realized there were some other opportunities for being a biologist. And I became interested in paleontology. So my master's degree project was to dig up about a ton of gravelly silt from a creek bank and sift through it with a little screen and some water and pick out all the little tiny teeth and bone fragments of the fossils that were there. And this was not too far from y'all. I guess around Aspermont is where this was, the Southern Panhandle. And we got a radiocarbon date of about 12,000 years. So the species that I was able to identify from the teeth and the bones and whatnot were whatever lived there in the Panhandle area about 12,000 years ago. And I found species that occur no further south than northern Nebraska, there in the Southern Panhandle. And so putting it all together, I inferred that the environment of the Southern Panhandle here in Texas was probably fairly similar to what it's like in northeastern Nebraska today. So that shows you how the environment can change. And then after that, I realized that there were not many jobs for paleontologists, and I became interested in genetics. And so I've been doing genetics ever since then. And as Matt mentioned, I met some of y'all about almost a year ago. And let me share my computer screen here so you don't have to look at me anymore. So this is an iNaturalist page of a pretty little flower that some of y'all may recognize this person right here. Glenda posted this. Now it's my understanding that Matt's the one that found the flower. Correct me if I'm wrong. And then Glenda posted some photographs of it. Hopefully I've given credit correctly there.
You're exactly right.
And so I have never really studied plants. I'm not a botanist. I've mostly focused on mammals. But friend of mine that I met through iNaturalist living in the Fort Worth area was interested in this particular flower. And so I got interested in it through her. And there's one species that is just a common yard weed where I live, and I never gave it a second glance. I just, little white flower down in the grass. And then I learned that there were more than one species of this yard weed in Texas, and that each one looks just a little bit different and lives in different habitats. So I learned to recognize some of them and started identifying people's observations. So people would photograph these from around the state, and I'd look at them and see if I could identify them to species. And I probably went through a few thousand images over a year or two. And so I sort of became familiar with what each species looked like. And in the county that I live, there's one species that's very common. But I found a record of a second species in the county that I live. Was collected by a former professor at Tarleton State University here where I am in the 1920s. And it has not been documented in this county since then. And so I saw that as a personal challenge, that I was determined that I was going to be the second person, the first person since 1920 to document this plant. I was afraid that it had been extirpated, locally extinct, because it only occurs in sandy soils, and there's just not a whole lot of sandy soil in this county. So I looked and looked everywhere, looked at the maps, looked where sandy soils are. Never did find it after few weeks of looking. One of my hobbies is to, that I share with my wife, is to explore local cemeteries out in the country. And so we were exploring a local cemetery about 1 mile from our house. And we were just looking along, looking at the headstones, and I noticed this little white flower down in the grass. And I bent down and looked at it, and darn it if it wasn't that plant that hadn't been seen since 1920, just a mile from my house. So that was fun. And then this pops up on iNaturalist, and Glenda had identified it as Anemone caroliniana, the Carolina anemone, because the flower looks just like the Carolina anemone, but they don't occur any further west than around the Fort Worth area. So I was like, why is the Carolina anemone in Lubbock? This is pretty crazy here. And so I got Glenda to post some additional photographs. So I told her to photograph some leaves, photograph the stems. And I was looking at these characteristics. It's so fuzzy and just has a very unusual growth habit. It's definitely not Anemone caroliniana from the flower down, but the flower does look like Anemone caroliniana. So I'm like, what is this thing? And I looked at it for a few days. Now this plant only blooms for about a month, and then you never see it again. It totally goes dormant for the rest of the year. So you basically got just a few weeks to see it. This was March 19th. And I don't know if y'all remember what was happening around March 19th, but it was called COVID, and the country was just about to shut everything down. So I gathered my wife and son together, and I said, there's this plant out at Lubbock, and if we don't go see it now, we're gonna have to wait a year to see it. Should we take the chance? There's a global pandemic. There's this plant. What should we do? So we gathered up a bunch of hand sanitizer, some 409 germ cleanser, booked a room at a hotel not far from Lubbock, and headed out. We were the only person in the hotel, which was pretty eerie. Next morning, got up and met Matt and Glenda in this little canyon, and just spent a really pleasant afternoon looking at, or it was morning, a really pleasant morning looking at these plants. I still don't know what they are. But this summer, I'm going to be trying to extract DNA from the plant along with some other species. There's 5 species that, ignore the bottom 2 here, just look at the top 5. This is the common yardweed on the top left. This is Caroliniana, the second one. Edwardsiana, the Edwards Plateau, grows only around the Edwards Plateau area. Okinonai is scattered around through West Texas, and then tuberosa is only found in Lubbock. So, not Lubbock, El Paso. Tuberosa found in El Paso. So where does the one that we found in Lubbock fit into this scheme of there being 5 species? That's what I'm trying to figure. So the topic of my presentation is the what is a species anyway? So when you find something that looks unique, you ask yourself this question: what is a species anyway? So how do I know if what I've discovered is a new species? So my working hypothesis is that it is a new species unknown to science. Is that wishful thinking? I don't know, but it sure doesn't look like any of the known 5 species. So I think there's a chance. Now, one of those 5 species was only discovered in the 1990s. So there might be a new species out there that nobody's noticed. So I'm gonna go through and talk about what a species is from a scientific perspective. And parts of this will get pretty technical because genetics is pretty technical. And if that's not your cup of tea, just put your fingers in your ear and close your eyes, and then come back, once I get through with the technical stuff. But I'll try to keep it fairly simple. So what is a species? There's 2 ways to think about what a species is. One's from a practical standpoint. How can you tell? By looking. And then one is from a theoretical standpoint. What types of things can be called a species? Okay, so you kind of have to think about both of them. What types of things do you want to call a species? And then how can we tell if any one organism belongs to one species or another? From the theoretical side, scientists don't agree. Scientists can have friendly and sometimes not so friendly debates over what things should be called species. So that's kind of what the presentation is about. From a practical standpoint and from a theoretical standpoint, what are these things that people are posting on iNaturalist and calling species? So we'll start out with the quiz for everyone, and you can either post your response in chat, or you can just say it out loud, or just say it to yourself. It doesn't matter. Here are some ladybug photographs. And so the question is, how many species are represented among that variation there? I'll let you think about it a few seconds. How many people said 1? Because that's a correct answer. So even though they look really different, all of those are individuals of the same species. And so that's what we would call phenotypic variation, differences in appearance. Here's another example. So here we have the common name. Whoops, I goofed, sorry. Here we have the common name and the scientific name, and then the year that it was described as a species. 2 different butterflies. But in fact, that's two sexes of the same species. The male is blue and the female is brown. But for many years, they were thought to be two different species. Here's a bird that lives, I think, in the Indian Ocean, if I remember correctly, on islands and whatnot. One species. Well, just recently, they were named to be 12 different species, each one very, 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 very slightly different. Some of them actually look the same. So we went from having one species one year, and now, hey, we recognize 12 different species of them. So what is a species anyway? Well, the practice of naming things is called taxonomy. And humans have been practicing taxonomy for as long as we have been pointing and grunting at things. So as long as we've had a language, we've been naming things. So all peoples around the planet, even the most primitive peoples far away from modern society, have names for things. Common names are not real good for science because they vary from region to region. So in the lower left, I would say that that's a gopher. And then on the right, or right-hand side, I would say that's a ground squirrel. But if I went to Florida, the one on the left would be called a salamander. To me, the salamander is the little amphibian there in the middle. But to Floridians, gophers are called salamanders. Probably the word salamander evolved from sandy mounders because gophers make mounds of sand. And if you got a wad of chewing tobacco in your mouth and you're saying sandy mounders, it kind of might come out as salamanders, and that's probably where the name salamander came from. If you're in North Dakota, what I would call a ground squirrel, there on the right, would be called the gopher. So that makes it really hard to communicate with people. So that's why we have scientific names. And so the scientific name of a species is a two-part name. The first part is the generic epithet, and the second part is the specific epithet. So this particular type of gopher is called Geomys bursarius. And everyone uses that name and doesn't get confused. So Latin is used because it's an extinct language. No one speaks Latin, and that's kind of makes it fair, about as fair as you can get for all of the different languages that people speak. Now, above the species level, we have families, order, class, file, kingdom, and domain. And so these are just different taxonomic ranks. And they are more and more inclusive as you go up. So there's many phyla within a kingdom, many classes within a phylum, and so forth and so on. And we just, us humans like to lump things and give things names. And this is just a product of being human. The groups that we lump things together in, we prefer them to be united by shared ancestry. So we want all of the species that we include in the order Carnivora, we want them to be related to one another, to be descended from a common ancestor, just like with their family trees. So when you put together your family tree, you want to trace it back to a shared great-great-great-grandparent that lived a long time ago, and everyone in the family is descended from that individual. You don't want to just pick people that vaguely resemble you and say, hey, you're in my family because you kind of look like me. That would be a strange way to put together a family tree. The alternative way of grouping things is just by shared features. So for example, we could group things that have wings or things that fly. So in a group of flying things, we would have bats and birds and pterosaurs and insects. But that would be a group that is not united by shared ancestry. So natural groups are what we are looking for. Natural group would be shared ancestry. So the thing is we don't really know for sure all the time what the shared ancestry is. And so we have to look at all kinds of data to try to figure out what that shared ancestry is. And then once we figure it out, we can modify our groups. So it's turned out that we've been wrong about our groups, and that many groups are not united by shared ancestry. They're just united by shared features, unrelated organisms in the same group. And whenever we discover that, we change our groups. So therefore, taxonomy changes as our knowledge changes. For example, when I took mammalogy and I learned about the different groups of mammals, well, that was 30 years ago, and we've learned a lot since then. And so now I really don't know any of the names of the groups of mammals anymore because they've been updated. And I'm going to skip this. I meant to delete that other bullet point there. I'm going to skip over that right there. Okay, so it's just a name. We put names to things, we make groups. So why does it all matter? There's 3 reasons why it's important to name things and to group things correctly. One is just to communicate with one another. So if I say, hey, let's all go out and look for a Carolina anemone. If I use the scientific name, then we all know exactly what we're talking about. To know what we're studying. So if you don't have names to things, you don't really know what you're studying because you haven't grouped them together and given them a name. And you can't tell other people what you're studying either. And then to know what to protect. So from a conservation standpoint, you need to have things grouped properly so that you can protect them, manage them as endangered and threatened species. So how do we define them? Again, what is a species anyway? Well, species are different kinds of things. But what kind of thing differs? The opinion differs. What kinds of things should we group together and call a species? And this has been debated for over 100 years. And the debate really hasn't slowed down a whole lot, although it seems to have died down a little bit. I'm going to talk about that debate and some examples of the results of that debate here in just a minute. So what scientists have come up with are species concepts. And these are the theoretical side. What kind of thing is a species? And then species delimitation. This is the practical side. What features do we look at to determine if an individual is one species or another species? So when I look at that photograph of the little anemone that Matt and Glenda observed out there at Lubbock, I'm having to consider both of these things. Theoretically, what is this thing? And then from a practical standpoint, what characters do I need to look at to convince other scientists that this might be a new species? Okay, you certainly don't have to read all of this. I'm just showing you this so that you can see that there are different theoretical ideas of what a species is. There's at least 26 different concepts of what a species is. Each one of these has its own advocate, its own person who invented the concept or is a proponent of that concept. So we have the isolation species concept, the recognition species concept, the the phonetic and genotypic cluster species concept. What are all these things? Well, each concept has its own properties, which are the things that you would look at to decide what species are. So you kind of have to choose your concept, and then from that concept, you make a decision on whether you've found a new species or not. So properties of species. Most species look different, but they don't always look different. So you can't use appearance as a property of species. Lions and tigers look different. No one would confuse them. But there are some other organisms that are definitely different species, but humans can't tell the difference. The animals themselves, the plants themselves, can recognize that they are different species, but we can't because they look the same to us. So then we turn to things like intrinsically reproductively isolated. What this means is that there's something about the internal workings of the organism that causes it to be unable to reproduce with other individuals. So an elephant, there's something about the elephant that makes it unable to reproduce with the giraffe. They're just internally, intrinsically, reproductively incompatible with one another. So whenever we see that, then you know for sure you've got a different species. But unless they're living together, you can't tell that, right? You're not going to know if they can reproduce with one another or not unless they have the opportunity to do so. And so that's not always useful. So then we can turn to a third strategy. They occupy different niches. A niche is just like a set of environmental variables, temperature, humidity, types of shelter, soil types, all those sorts of variables out there in the environment. So it's usually the case, but not always, that different species occupy different niches. So one species might prefer a more humid environment, another species might prefer a more dry environment. But they might look the same, they might look different. And then this fourth one is a little more technical, monophyletic. This means they're descended from a shared ancestor. And we usually want our species to be descended from a shared ancestor just because, we want family groups to have that same characteristic. So then we have to decide, well, should a species have all of these properties or is just one or two of them okay? Would that be sufficient? Now, since monophyletic is a little bit more complicated to understand, let me describe this because I'm going to be showing these branching diagrams when I get to the section where I show you about my own research. So down here at the bottom of this tree-like diagram, and we call these trees, you have this single stem or trunk, and we call that the root of the tree. And then that branches out to form two branches, and then each of the two branches forms branches. So it's very much just like a tree-like structure. And so over here on the right, where you have the red, this is indicating that each of these individuals, so each of the tips across here is an individual organism. And so each of these individuals is connected together on a single stem. And so this single stem would represent shared ancestry. They all branched out, descended from that shared ancestor. And then here, the green and the blue, they all branch out from this ancestor down here towards the bottom there. And all of the blue ones, they branch together and diversified from that shared ancestor there. Now, so we could use this pattern of relationships, pattern of shared ancestry, to say, well, all of them are the same species. So we could say that's one species because they all group together here. Or we could say there's two species. The blue and green makes one species because they all group together down there at the bottom left. And the red, that's a second species because they all group together here. Okay, so nested groupings. This is how we determine if they have a shared ancestor or not. And we can use morphology, or what I do is I use DNA sequences, DNA data, genetic data, to figure out these patterns of relationships, because DNA is handed down from one generation to the next. And so it reveals those ancestors. So how do species arise? So down here at the bottom of this diagram, we have one single species that existed in the past. No longer exists now, but it's, maybe 1 million, 2-3 million years ago, there was an ancestral species, and that there was some barrier to genetic information moving between two populations. Now, this barrier could have been a river, could be the formation of a desert, could be that that a continent split in two, and now you have an ocean that separates two continents. So I'll use that as my example. So let's say this ancestral species was occupying a continent that split into two continents separated by an ocean. And so the genetic information is not going to be able to go from one population to another. Over time, mutations occur. Changes in the DNA sequence are going to happen in both of the two populations. Natural selection will occur. Random fluctuations will occur, causing each of the two populations to become genetically different. So every generation acquires new mutations, even in humans. So if you've had kids, you passed on about 50 brand new mutations to your kids that never occurred before. And your grandkids will get 50 brand new mutations that their parents didn't have. And so if you envision these two populations on different continents over 1 million generations with 50 mutations each generation, you're talking about a lot of genetic differences accumulate over that period of time. And so today, we would then see those as two different species because they've become so different genetically. And this is just showing some different species concepts and the associated biological evidence there on the right-hand side. And some of this is too technical, so I'll just look at some of the simpler things. So let's say that some of those genetic changes cause them to look different, morphologically distinct. They look different. Maybe if you're talking about a flower, different number of petals, different color of petals, different shape of a leaf. And so we would then say, based on the morphological species concept, they're different enough to be called species. And then more time passes, more mutations accumulate, and they come to occupy different habitats. One maybe lives in a drier area, one in a more moist area. So then we could say, based on the ecological species concept, they live in different habitats, so they're unique species because of that. But maybe they still can reproduce with one another. So it's not, a few thousand generations later, they accumulate more mutations. And now finally they've become reproductively isolated and they can no longer reproduce with one another. So they fit the biological species concept. But they're not yet monophyletic. They don't form these groups in the tree diagrams. But given a few thousand more years of mutations, they might eventually reach that state as well. So the different ways of recognizing species actually accumulate over time. And that's the That's the big idea. This is the central idea of what a species is. The order in which these different properties that we use to recognize species, the order in which these occur varies. Some species might become reproductively isolated before they look different. That makes it tricky because you can't just go out and collect them and observe them and realize they're different species. You might have to sequence their DNA to see that they're actually different. So there's no rule, there's no law as to which genetic changes occur first. And so scientists get confused over which species concept is the most important. Well, none of them are the most important. They're all useful in their own unique ways. So when I'm looking at that anemone plant out there in Lubbock, comparing it with the other species, I'm looking at all these different characteristics to try to see which boxes I can check. And the more boxes I can check, the more confident I am in calling it a new species. Now that describes speciation over millions of years. Speciation can occur almost overnight, over a very short period of time, by changes in chromosome number. So chromosomes are these little bundles of DNA that are found in our cells. Most people have 46 chromosomes, but some have 45 and some have 48. There's some variation there. This is showing the origin of multiple species of wheat by changes in chromosome number plus hybridization. So there's two species of wild wheat here at the top right, two species of wild wheat, and their chromosome number changed and they hybridized to create this wheat, which has twice the number of chromosomes. This species of wheat has chromosomes from both of those species, A and B. Notice it's got chromosomes from both species. And then that species changed its chromosome number by hybridizing with a third species. And so now we have wheat that has 3 different sets of chromosomes from 3 different species. And this is the wheat that we plant and harvest for our bread. It has not 2 sets of chromosomes like what humans have. It has 1, 2, 3, 4, 5, 6. It has 6 sets of chromosomes from 3 different species. And that all took place in about 8,000 years. Okay, so speciation can be relatively quick or relatively slow. Okay, so I'm going to pause here. The next thing I'm going to do is talk about my research and how that fits into the problem of what species are. So I'm going to pause and are there any questions that people have before I move on? Okay. Inside a cell, there are all kinds of little compartments. And some of these compartments contain DNA. The nucleus contains most of the DNA. And if you, I'm not sure how familiar all of y'all are with genetics and DNA, but I'm sure that you've heard that DNA is made up of G's, A's, T's, and C's. And inside the nucleus of our cells, we have 6,000 G's, A's, T's, and C's. Not 6,000, 6 billion. That's quite a bit more. 6 billion G's, A's, T's, and C's. We got 3 billion from our mom, and we got 3 billion from our dad. That's all in the nucleus. But we've got DNA in these little compartments called mitochondria, the powerhouse of the cell. There's DNA in there too. but only about 16,000 G's, A's, T's, and C's. So most of the DNA is in the nucleus, but we do have DNA in the mitochondria as well. We only get our mitochondria from our moms. The mitochondria in all of our cells comes only from our mother, never from our father. But the DNA in the nucleus comes from both parents. And because of that difference, we can kind of understand ancestry a little bit better. Okay, so now going back to iNaturalist. So during the upcoming City Nature Challenge, y'all are going to be going out and photographing and uploading those photographs to iNaturalist. You're going to find as many different things as you can possibly find. And they're going to be plotted onto a map that you'll be able to see. And then everybody else can see those as well. And so people like me come along and they can look at these observations that people like y'all post and learn some really interesting things about the organisms. So I mentioned that I've studied mammals, fossil mammals, but then also modern day mammals and genetics. And I've recently gotten interested in a plant. And I've also recently gotten interested in the bumblebees. Prior to about 3 years ago, a bumblebee was just this black and yellow bee that stings really bad if you make it angry. Just recently, I've learned that there's 2 different kinds of bumblebees right here in Texas. In fact, there's 2 different kinds of bumblebees in the county in which I live. So because I'm interested in different species, that sort of piqued my curiosity. And it really got interesting when I learned that some people think that these 2 bumblebees are the same species. And so I started looking, I started identifying bumblebees on iNaturalist, and I identified, I don't know, maybe around 5,000 bumblebees over the past couple of years on iNaturalist. And so, well, once you look at several thousand photographs of 2 different kinds of bumblebees, you get a pretty good feel for what they look like. And I'm thinking, these 2 bumblebees look really different, and they're consistently different. I never see anything that looks like a mixture between the 2. So look at the 2 pictures here and see what you think. Do they look the same or do they look different? So if you look at the top one, the color of yellow is more vivid than the one at the bottom, which is more of a pale pastel yellow. The one at the top has 2 bands of yellow by the wings, and the one at the bottom only has 1 band of yellow by the wings. So there's those subtle characteristics that you start to pick up on once you look at a few of them. And I read in the scientific literature where people thought they're the same species, just different varieties. Because somebody had seen the male of the Sonoran bumblebee at the entrance to a nest of a female Pennsylvania bumblebee. And so because of that, they thought, well, they're probably reproducing with one another. And I'm looking at these and I'm like, I don't see any evidence of hybridization. I don't see any evidence of one species mating with another. And so I decided that I'm gonna prove once and for all whether these are the same species or 2 different species. So I collaborated with a couple of folks at another university, and they provided some bumblebees, and I sequenced the DNA. And here's my little tree that I created from the DNA. So each one of these that's labeled DF, that's 1 bumblebee. And because they're all together on this 1 branch, they basically are genetically identical. But then they're all on this long branch that connects way back here to these other bumblebees. So each one of these is a different bumblebee. And it turns out all of these are the Pennsylvanicus form, and all of these are the Sonorus form. that you never see them intermingled in the same branch. So that means that they fit the monophyletic criteria. That's evidence that they're different species. Genetically, they're all unique. So that's a project that I'm still working on. So I'm collecting bumblebees here around my house because both forms occur here locally. And so I'm going to do some additional genetic and morphological work. A few months ago, these were petitioned to be on the endangered species list because they're declining in some parts of the country. In California, in New England, these species are becoming much more rare. And so knowing whether they're one species or two was going to have some practical value, I think. So if you don't like bumblebees, maybe fish. So I've worked on a fish project. So there's these two types of fish, which they're minnows, about the length of your finger, the black-tailed shiner at the top and the red-finned shiner at the bottom. I grew up seining creeks, catching these to put on the trot line. So it's kind of neat to now be a geneticist them. A publication, a research paper published a few years ago, documented that in Georgia, these two species hybridize extensively. But in Georgia, only the top one is native. The bottom one, the redfin shiner, has been introduced recently to Georgia by. So the question that I have is, are they hybridizing extensively here in Texas? Because species are native to Texas. So here's what we did. So I live in Stephenville. That's this town right here. Hopefully y'all can see my pointer. I hope y'all can. So there's Stephenville. And there's these two rivers, the Paluxy River and the Bosque River, in which both species can be found. So we sampled at 4 places along both rivers, caught the fish, extracted the DNA from the fin tissues. And this diagram in the upper left is the results of the genetic analysis. What the heck does this mean? Well, the little white circles down at the bottom, each circle is an individual fish. And if it's white and close to the bottom, it's a black-tailed shiner. If it's a black circle and up at the top, it's a red-finned shiner. And so each location maps out here. So we found the black-tailed shiner at every site, but at the downstream site, P4, we found mainly the redfin shiner and not so many black-tailed shiners. So they kind of discriminate by habitat a little bit. They seem to prefer different habitats, upstream and downstream. Hybrids would fall at the 0.5 line. So we didn't find any hybrids at all, but they don't really occur together. You know, the redfin shiner is kind of downstream and the black-tailed shiner is upstream. So then let's go over to the Bosque River and see what we find here. So we found a little bit different pattern in the Bosque River. So again, the white at the bottom is the black fin shiner, the black at the top is the red fin shiner, and anything in the middle, if it was at 0.5, that would be a hybrid between the two. If the two mated and produced offspring, it would be 0.5. It would have half the DNA from one and half the DNA from the other. So we didn't find any hybrids. We found, maybe this is the closest thing to a hybrid we found right there, but that's just one. And in the Bosque River, we find both species occurring together up and down the river. So even though they're occurring together in the same river, they don't appear to be hybridizing hardly at all. Definitely not extensively like in Georgia. So something weird's going on here. So what we think might be happening is here in Texas, where the two fish have lived together for thousands and thousands of years, they've learned to recognize one another as different species. They've developed intrinsic reproductive barriers that prevents them from mating with one another. But in Georgia, where the black-tailed shiners have never, ever, ever seen a red-fin shiner, they don't have that genetically built-in mechanism to recognize them as a different species. And they're like, sure, we'll mate with you. And so they hybridize extensively. So you can have weird things like that going on, where one population of a species is unable to mate with another species, but another population of the same species is able to mate with another species. So that becomes really confusing. And then here's another project that I've done. There's this cute little rodent called the cotton rat. Y'all have them up there in the Lubbock area. It's one of the few rodents that you'll sometimes see scurrying around during the daytime if you have thick vegetation. And sometimes they'll come up to a bird feeder and eat the seeds below the bird feeder. And somebody had discovered that there's a little bit of difference between the east and the west forms. So here's where they're found in the United States. And so we decided that we'd go out towards West Texas and run a transect. So we trapped cotton rats at each of these 10 locations across these 4 counties in East Texas. The tissues from the organism and did some genetic work. And here's what we found. So we took some from Kansas, Oklahoma, different locations here. And so we have Kansas, Oklahoma, and Texas right there. That's represented. Took some from Louisiana and Georgia, and those are representative of the eastern form. And then there's our 10 sites along our transect. So each bar represents the genetic information from one individual cotton rat. And if it's solid gray, up and down, if it's solid gray, that means it's basically a pure Western cotton rat. And if it's solid white up and down, like this one here, that would mean it's a pure Eastern form. If it means it's a Western that mated with an Eastern. We really didn't see any of those. There's no vertical lines that are solid black. And F2 would be if 2 F1s mated with one another. It's another type of hybrid. The back crosses occurs when you have a hybrid that mates with a pure. So an Eastern back cross would be a hybrid that mates with an Eastern. And a Western back cross would be a hybrid that mates with a pure Western. And that's kind of what we saw. So at sites 2, 3, 4, and 5, we saw those cotton rats are mainly hybrids that mated with the Western. And then on location 6 and 7, we found that most of them were hybrids that mated with the Eastern, but there's also some F2 hybrids in there as well. So we were able to sort of capture the hybrid zone between 2 genetically different forms of this one particular species. So since they're hybridizing, they are reproductively compatible with one another. And since the hybrids are mating, it sort of forms a continuum of genetic variation. It blends together from pure Eastern to pure Western with hybrids in between. So are they 2 different species that hybridize or 2 different varieties that of the same species? It's up for debate. And I think this is the last example here that I'll give. I'm really interested in gophers. Y'all got a couple of different species of gophers out there in the panhandle. Someday I might get out there and collect some gophers from the southern panhandle area where there's an interesting hybrid zone there. but these are from the El Paso region and New Mexico. And there's two different subspecies, which is two different sort of varieties of the same species. One's in the northern New Mexico, one's in southern New Mexico and Texas. Lubbock is not in the Panhandle. I am, yes, that's probably a very offensive statement that I made there. So what we did is we collected specimens from several different places here in New Mexico and Texas, and created a tree diagram based on the DNA sequence. And we found that all of the ones, this is supposed to be color coordinated, I apologize. These are supposed to be the orange group up here, and then these are the green group down here. So the two different groups did form genetic groups as well. So we verified what some people back in the 1980s suspected based on other characteristics. So we definitely have two distinct groups of this one particular species out there. And if we look at, so that's based on the DNA in the mitochondria. If we look at the DNA based in the nucleus, we see something even more interesting. So each symbol is one gopher, and it shows where they're collected from. So the green circle, these gophers are from this area down here. And the ones on the left-hand side of the diagram, they are in the orange region up there. So we separate those two forms again here, but within the orange group, we find two distinct groups as well. So the groups in the far north are genetically different than those further south here. And so we actually have three genetically different groups of pocket gophers. So is that one species? Is that two species? Or do we have three species? Again, that's subject for debate. So then we come full circle to try to address that question again. So what is a species? Well, we take into account those theoretical and practical issues, and we come to this conclusion that people have been arguing over what actually is a species for over 100 years. That's what we call the species problem. Nobody has come to an agreement over what a species actually is. And so scientists have been debating this for over 100 years. It turns out Mother Nature created the species problem. So it's not a problem that humans can solve. We cannot define our way out of it. It is just a problem that exists, and we just acknowledge it and move on. So species is one point along a continuum of divergence. So when you have population that becomes split into two populations, they over time become different varieties. And with more genetic changes, they become different subspecies. And with more genetic changes, they become different species. So we have these different sort of categories for how we divide things up. And it's based on how long they've been diverging from a common ancestor. So all those species properties that I've been talking about with the examples that I've used and everything, those arise cumulatively over time. And if enough time has passed, they become distinct enough that humans are compelled to say, yes, they're distinct species. Sometimes they're in the gray zone. So when you have varieties or subspecies, we can have friendly debates about whether those are should be recognized as species or not. And so as you're out there documenting biological diversity for the iNaturalist City Nature Challenge, and hopefully all of you will be doing that, as you're documenting things, think about just how complicated all this is. And much of that probably didn't make a whole lot of sense, but hopefully it gives you a vague idea of what scientists are doing behind the scenes to try to categorize the diversity of life that's out there, so that we can all understand it, talk about it with one another, and hopefully enjoy it a little bit better when we realize there's all these little subtle details out there that have, in many cases, yet to be discovered. Okay, that's all I have. Open the floor for questions. And
Anyone
it
have-
can be simple stuff, it can be simple stuff like, how do we use iNaturalist to recognize species, or more complex things.
I found your presentation fascinating. It's been 50 years since I sat in a classroom and studied biology. And to see how far things have gone and have come, I don't know that I could have passed the class in the modern day. It's seriously fascinating. What you do and what people like you do just amazes me. And I thank you so much for your service to society.
Russell, how long have you been doing iNat now?
That is a good question. Let's I wanted to kind of explore iNat just a little bit here. So let me look. My observations. The neat thing about iNaturalist is it's a record of things you've seen, things that you've found interesting.
Mhm.
And if I sort them by date observed, I find that
Ouch.
I haven't been doing it since 1972. That's an older specimen. I started observing things in iNaturalist 2012. So I've been doing it since 2012. And I currently have seen, I've currently posted only 8,483 observations. Someone like Sam Keeshnith that spoke to y'all last time, he's got way more than that. But it's really neat to think back that since 2012, I personally have seen and photographed 2,618 different species of living organisms.
Wow, that's amazing.
It's really amazing how many different species that you can see over such a short period of time if you're, dedicated and diligent and out there looking.
Do you find yourself referring back to things that you recorded back when and kind of referring back to it? I find I do that. I look back at things, something will pop up and I'll think back. And so I think it is a helpful tool.
Definitely, because I don't have a very good memory when it comes to names of things, which is a little ironic since I study naming things and whatnot. I can't remember people's names, nor can I remember names of bugs or plants. And so I definitely refer back to it a lot.
Dr. Pfao, this has been absolutely fascinating, but I do want to ask, since DNA is the baseline for differentiating species, do you have any idea how many total species have had the DNA examined? And of course, that's awfully broad. I mean, plants,
Wish I
fungi.
did, 'cause that would be an interesting number to know. It would be a tiny fraction of the species that have been described, because we've only been doing genetic work for such a short period of time, the past 20 years or so. And there's millions of species of beetles, for example. And I would say that probably only 1% of them at most of just beetles have been examined genetically.
Wow.
So there's probably a lot more species out there, because usually when you start looking at the genetics, you discover what we call cryptic species, which are species that look the same, but genetically are actually very different. And the organisms themselves may recognize one another as being different. There's a neat caterpillar that people used to think was just one species. And people started noticing that the butterfly looked the same, but the caterpillars looked a little different, and they fed on different plants. And they started looking at the DNA and found that, lo and behold, there are like 6 different species of them. And that's only just in the past few years that we've discovered that. So it's been pretty neat.
You know, in part of your presentation, you were talking about a bird, I think it's maybe in the Indian Ocean or in the Far East somewhere. And
Mhm.
one species, now it's 12 or so. Do you find the converse of that, like with juncos, where there were many described and you would put in, you know, there's have this color or that color, and now there's one, you just say you have a junco. Do you find that often?
Mhm.
Really? Interesting.
Yeah, I would say probably I would say in the past 20 years or so, we've probably been splitting things up into multiple species more often than lumping them together.
Hmm. Okay.
But we do find instances of both. Yep.
Yeah, interesting. You were speaking about the anemones earlier and saying that one of the 5 has not been seen since 1920. Is that, did I understand that?
Only in the county that I live in.
Ah, okay. I
Yep.
see.
It's much common in east of here.
Okay. How long have you lived in the Stephenville area?
Since 2000.
2000.
So 20 going on 21 years.
Wow. Well, my family, my mother's side, Gatesville is where they're from, and that's what, 50, 60, 70 miles south.
Mhm.
Beautiful part of the state.
Yep. Mhm.
Presentation was fascinating. Does anyone have any additional questions or comments?
One thing, Tarleton State sure seems to have a presence in biology. Is that, you know, from my impression is that's a college you really could recommend to any young person wanting to study biology.
Charleston's fairly unique, and I don't really know why. The enrollment is been steady or declining in several universities in the state, but Charlton's enrollment has been going up and up and up. When I got here 20 years ago, we had, I think, around 5,000 students, and now we're up to around 15,000 just in 20 years. So somebody's doing something right, I guess. So it's been fun to watch Tarleton kind of grow up from being a community college to being a more larger, more research focused. And we do our best to involve our students in the research that we do. So pretty much every faculty member that's doing research has 2 or 3 students that are doing research right along beside them.
Wow. Well,
So all of the projects that I talked about there, all of those involve students doing field work and doing the laboratory work. So they get to learn all those skills involved and take that on into their careers.
Are most of these undergrad students or?
Probably more undergrad students than graduate students are involved in research.
It has to be pretty exciting for them.
You know, one of my current students has just been accepted into a PhD program at 3 different universities. So she's having to make the tough choice of which university to go to.
That's cool. Well, if you get a chance, come see us.
Okay, will do.
Thank you.
Y'all keep an eye out for those anemones.
We will.
We will.
It's kind of
Thank
fun.
you. Thank you for presenting, and thank you for sharing some of the genetic information. I especially enjoyed that. Thank you so much, Russell.
Yeah, really nice, Russell. Thank you so much. And
Yep.
thank you everyone
Thank
who
y'all.
signed in to listen this evening.
Thank you.
Thank you.
If
Thank
there are
you.
Bye.
no
Take
further,
care.
Bye-bye.
Bye-bye.
thank you, Russell.
Bye-bye. Thank you.
Appreciate it.
Everybody, thank y'all.